Hello. In last few sessions we have understood the nature of feedstocks, how these feedstocks are very important, and before tapping them out you must know the other components like its feasibility factors, like in terms of economics, in terms of yield, and the very important thing is that sustainability. So these sustainability factors are very important things, parameters in this particular domain, that is biofuel. Biofuel basically depending upon two segments. One is bioresource and another one is the technology. That is why this one today's session is very much based on this bioresource technology. Means the resources those are available in nature need to be understood very carefully and holistically. Only then you can employ the things of technology. Things means you have to understand the technological input as well as output. Herein, this bioresource, this is very much constant. This bioresource is very much constant, whether you talk about wheat husk, you talk about sugar cane, you talk about paddy husk, or any forest based lignocellulosic substrates. You must know this thing. Means the component which is very much in giant in shape available in the bioresource pocket, that is cellulose. Then hemicellulose. These cellulose must be understood biochemically, because if you are failing to understand its giant structure and function, how it looks, how it conserves energy, in which form it conserves energy, until unless knowing all these facts, you cannot proceed with your technological inputs. That is why people are doing this bioresource, you can say, characterization with the 3D manner, means structurally as well as the functionally, wherein the instrumentations like people are using NMR, so many other spectra, Raman spectra, multitops, n number of instrumentation facility is available nowadays. But depending upon your interest, depending upon your desirous goal, what kind of product you are bringing into the market at the industry level, so that your industrialist, those are going to be your ultimate beneficiary, will be interested. When they will be interested, why they will purchase your you can say monopolized patent, why they will explore the published data, so that you can further proceed for the patent and other kind of the commercialization. You need to understand all these things very carefully. Nowadays n number of research papers are available, and based on these research papers you cannot, you can say, conclude your roadmap, you cannot define your roadmap until unless you are not developing your own input parameter and the desirous output. That is why I have termed this bioresource technology for this particular theme. So let us see about the today's I have already connected you, I have already explained you the information at the generation level, where we are standing right now. We are very much in these two stages, second generation and the third generation. People are very much, you can say, progressives, very much, and doing good progress at the industry level, and countries, world is being reliant upon these things. Just like varieties of carbohydrates, what is ultimately this bioresource means? What nature is offering to you? That is very important. It is having immense concentration of the sugar, you must be knowing. It is having starch, it is having cellulose, hemicellulose. So you have to be very good student first of all at the biochemistry level. Only then you can synthesize and fabricate the things slowly. And of course I am tapping the young generations, those are very much beginner at the technological level. So basically these are the pockets of the carbohydrates. Carbohydrates are polyhydroxy aldehydes. You must be knowing polyhydroxy ketones or compounds that can be hydrated or hydrolyzed from n number of the means. So the smallest carbohydrate is a glucose and fructose that cannot be hydrolyzed to small carbohydrates. Units are called monosaccharides. This is the unit of the, you can say, energy pocket, monosaccharide. It means it will take least you can say kind of the energy to be further processed. That is why people are just disseminating power and then grabbing the monosaccharides from this complicated complex molecules. So those consisting of same two monosaccharides like lactose and maltose or different compounds are called disaccharides. Similarly, you can go up to the polysaccharides, means NNN. So that is why this is the ABCD level of the information. You need to begin with that. And then primary plant monosaccharides, you must be knowing D-glucose, L-glucose, D-xylose, all these things, dextrose and all these things, right? So D-glucose serves as readily available chemical energy and as a supply of carbon. Because why we are understanding these things? Because the nature is having the complex substrates. I have already talked about the cell wall complex. Cell wall, you can say polymers, biopolymers. Those are having the big molecules like cellulose, like hemicellulose. But these are very much wrapped with the other kind of the, you can say, defensive molecules, that is lignin, because lignin is providing the recalcitrancy to the plant cell wall. So that is the kind of the big nut. You need to crack that bottleneck, and of course it is bad for us but good for the plant system for their existence. So how come how you will deal with that? That is the interesting part of this journey. We need to understand. This is the D-xylose. Of course, you have to understand a little bit the chemistry, how many carbon molecules are there, how many hydroxyl, you can say, bonds are there, whether it is covalent or you can say non-covalents, and what kind of equivalency they make. So these are the kind of the associated discussions you have gone through at the +2 level. So these two sugars in combination with several other minor sugars serve as the building blocks for the productions of more complex carbohydrates. So you need to understand, and I hope you are understanding all these things. Amylose, similarly you have to understand these structures. So I'm not taking your much time to understand all these things. Basic chemistry, lignocellulosic biomass. I have already mentioned what is lignocellulosic, means lignin plus cellulose. When these two are sitting together, then forming the rigidity at the plant cell wall level, and that is why it is having the kind of eractum. So lignocellulosic biomass chemically consists of three basic polymers: cellulose, hemicellulose, lignin bark. These are the three important candidate biopolymers. These are playing biggest role at the cell wall level, and that is why we are very much interested to tap all these things. So it looks like this. A structure of lignocellulose. See, comeryl alcohol, coniferyl alcohol, synapyl alcohol, all these things, and then this is your plant, and plant cell look like this, and the cell wall is there, and if you are portraying these things through the kind of the pictorial explanation, then you'll find the cellulose at the macrofibril level, and macrofibril means it is a giant cell. So how much it is connected to each other, that is why it is providing the rigidity and the strength to the plant cell wall. So it is having the macrofibril 10 to 20 nanometer. Why we are discussing all these things? Because ultimately you have to evade this kind of the structure, and only then you can harvest your energy. And when you will harvest the energy, then you will find in this manner. See, they are very much linear, and they are very much in the kind of the umbrella like of the things, and wherein the lignin is also very much wrapped around the, you can say, macrofibrils of the cellulose. So what is the abundancy factor here? This composition of lignocellulosic biomass. Lignocellulosic material, so world's largest bioethanol renewable resource. How much it is offering? Cellulose is offering 30 to 50% of the total biomass. Hemicellulose is again second number, standing second, 15 to 35% biomass. Lignin 10 to 25% biomass. So that is why we are very much interested in tapping the cellulose and hemicellulose. Those are make up approximately 70% of the entire biomass, right? So now the story begins with these things. This is the distribution chart of the composition of lignocellulose, hardwood. These are the materials, stocks, bioresources, candidate feedstocks, hardwood stems having 40 to 55%. These are all theoretical parts. You can memorize it, you can note down it, you can find it from the sources. Cellulose, you have to understand, you have to create the proximity with this thing. Cellulose is a homopolysaccharide, means like-minded saccharides are sitting together. That is why it is very much multiplied by N, and which is composed of beta D glucopyronase units linked together. That's why it is very much powerful, charged 1 to 4 directs, 1 to 1 dies to 1 prime to 4 prime glycosidic bonds, which consist of approximately 40 to 50 weight percentage of dry wood and provides wood's strength. That is why because of this kind of the structural orientation, cellulose is the most powerful bioresource molecule, biopolymer. After removal of water from each glucose molecule, glucose anhydride long cellulose chains containing 5,000 to 10,000 glucose units are formed, namely cellobiose units. So you can understand how people are segregating and finding the smallest chap from the complex molecules. Like this, structure of cellulose is very much like this: non-reducing ends group and the reducing end group, because it is having the OH. Here also having the OH, but you have to understand the functionality or functional group at the prime level. You can say, if you understand the gene a little bit, then upstream, and this is the downstream reducing end group. So I'm not going to repeat this again. This is also very important, lignin, because see, lignin is just protecting. Lignin is giving the coverage to this powerful energy system. So it is not discouraging you, though it is giving the kind of the message, okay, once you are done with the harvesting process, then how you will cover them. Of course, whether it is a hydrous or not anhydrous, you have to understand these things. So lignin, a phenyl propane based polymer, is the largest non-carbohydrate fraction of lignocellulose. You have seen cellulose could be digested, cellulose could be diluted in water, but that is why this lignin, you can say anhydrous non-carbohydrate fractions of lignocellulose, is sitting there with its phenyl propane based polymer. The main function of lignin is the support through strengthening of wood, like that pumping cells, xylem and phloem. That is very important. So lignin is indigenous. Lignin is indigenous, and its structure, it's very giant, huge structure it's had. So you have to see, if you have time, or certainly I will come up with the lignin biosynthetic pathway once in the separate story. So basic question is here, how you will produce. Before coming to this, just have a little bit glance of this thing, starch versus cellulose. Just you must be knowing both of them are very much sitting together and very much close to each other as far as stereochemistry is concerned. Monomer, starch C, glucose, alpha 1,4,1,6 linkages, dimer sugar maltose used for storage. So these are the list of starch coming under the name of the starch. These are the cellulose, glucose, beta 1,4, cellobiose. So what you are saying? You are saying how you are fabricating these two components. It is coming in the form of granules at the end, in the coil form, bending form. It is landing with your long fibers, like chain. So this is the basic, you can say, message of this particular presentation. As far as now, these two important candidates, cellulose versus hemicellulose, it is having monomer, is a pure glucose, it is mixed glucose. Because see, if you see the nature of hemicellulose and the structure of hemicellulose, it is very much linear. For example, xylene chain, you can mention the xylene chain under hemicellulose. You'll find a very simple structure as compared to the cellulose, because you can see its longevity or long, how much, 5 micrometer. It is very much short, it is very much high units, having 1000 units per coil. It is very low, 100 units per coil. It is very much linear, but it is very much branched, so that you can prime it as and when you require. So no substitution it requires, but it has a substitution C2, C3 and C6. So that is why hemicellulose are being considered as a brilliant molecules as compared to the cellulose. But cellulose is sitting the giant club, and hemicellulose is having very free candidature, so that you can accept it, and you can saccharide it, and you can say process it. That is very important. This is the pathways of ethanol production process. For example, you have taken corn and you have taken a different process to digest it, because digestion is very important thing, where you are sacrificing more than 70% of the energy available in this particular feedstock while processing. For example, you are doing cleaning, you are doing miling, you are having the liquefaction, saccharifications, means you are fabricating it, just like you have taken the cotton ball and then you are just fabricating it, then you are making kind of the small ball, and or you can proceed with that in a linear manner. So that is why in due course of doing these things, you are sacrificing a lot of energy. As well, it is not very simple. People are still hunting for the simplest pathways. So this is the established pathways as of now at the industry level. People are following any pathways based on your kind of the feedstock. So here feedstock is corn, and in both the ways this corn feedstock are being dealt. Ultimately this intermediary, you can say enzymes, alpha amylase and glucoamylase. These are very much involved in bidirectional ways and involved in liquefations. Means now it is digesting. So starch is being digested, right? And then followed by the saccharification, where you can use the very important domain, that is fungus, yeast, Saccharomyces cerevisiae. And yeast is fermenting and then producing byproduct carbon dioxide. And then again it is being distilled, and then dehydration, then ethanol is with you. So this is the shortest. I have chewed up so many steps in between, and trying to explain you how you are you can say collecting your ethanol at the end. So basically you can see you have taken the corn feedstock, then you have followed this protocol by dry miling or wet miling. You have cleaned it, then again you have miled it through the milling process, and here this is a little bit exhausted process, cleaning, then steeping, then degermination, corn oil, germ meal. So people are also, if being economical and very much with a business mind, people are harvesting other byproducts also in due course of these things. So people are making corn oils also, fiber also being used by N. Then people have got the gluten separations, and then got starch, then liquefations, then saccharification, fermentation with the help of the microbes, where you can use either yeast, or you can say Pichia pasteuris, or you can even use the microbes like E. coli. Then distillation, then dehydration. So this is the pathway as of now I have taken into the discussion. So for today's, just I have given you the wholesome journey, how you begin with and how you need to be a very good, you can say, researcher into this kind of the field. Thank you so much.